step that we have here is forming the peptide bond. And what's amazing about this is it occurs right in the middle on the very inside of the ribosome. There's no protein around there at all. And so the catalysis is carried out exclusively by RNA, side chains, RNA bases. And so this upset the dogma that we had before that proteins always carried out enzymatic reactions, and RNA and DNA were just for information. Here we have a case where the RNA is actually forming the active site where the catalytic reaction occurs. The type of reaction that occurs here is the amino group on the amino acid transfer RNA at the A site nucleophilically attacks the peptide bond at the attachment to the transfer RNA, which is an ester linkage, forms the new peptide bond that you see right there, and this transfer RNA in the P site is now unloaded. It just has an OH. So we've now attached the peptide in the P site to what is now the A site, occurring in the very middle of that ribosome complex. Here again is another picture of that type of reaction. Again, the amino group nucleophilically attacks that phosphoester linkage and forms the new peptide bond that you see right there. And the thing that assists this is a piece of the RNA, and that particular base acts, as you can see, as a general base, accepting the proton, allowing this amino group to be a better nucleophile in carrying out that attack. So that's how the mechanism is thought to occur. Here's a little more detail. It's thought that we go through a tetrahedral intermediate. So the amino group first attacks the phosphodiester bond here. Then we have this tetrahedral intermediate, and then a proton comes in. We break that bond, attach the proton to that empty transfer RNA, and we have the new peptide bond, which is colored green here for the new amino acid. So that's the new peptide bond that we've synthesized right there. So that is the peptide bond formation step. That does not require energy directly. The energy was required in the loading of the transfer RNA into the A site, but the actual peptide bond formation doesn't need extra energy. It's actually provided by this activated form of the attachment of the amino acid to the transfer RNA right there. So that's really what provides the energy. Okay, this then is going to be this translocation step, and this involves another translocation factor, EFG, and this is a critical step because we have to move the messenger RNA together with these two transfer RNAs by one codon to the left. Or if you think of the messenger RNA as stable, you move the ribosome by one codon to the right. And this has to be absolutely specific. There can be no slippage in that interaction between anticodon and codon. If they're off by just one nucleotide, that will make a fatal error in the whole thing. So the group that does this is elongation factor G. It binds into that site next to the A site. It hydrolyzes the GTP to GDP, and it just shoves the whole thing by one codon to the left. It just shoves the whole thing, and so that's pretty amazing. Now, let's look at the structure of this EFG, and it looks quite a bit like a transfer RNA in complex with elongation factor TU. It looks like a corkscrew, but this is all protein. In the TU transfer RNA, that thing that looked a little like that was transfer RNA, but that's all protein. So that's the different parts of the EFG complex, and the GTP is binding way up there. So that's where the energy is going to come from. So again, these people that I listed that received the Nobel Prize have got some nice X-ray crystal structures. here is where the EFG binds, right in there. And it binds much the same way as the transfer RNA TU complex binds. And once it binds in there, we are then going to get this huge conformational change occurring. And these are thought to be the steps in this. So what we have here is we start out with the two things in the P site and the A site. We then shift the top of them over to the P and the E site. So that's a preliminary shift. Then the G binds. Then we hydrolyze GTP. And while that hydrolysis is going on, we shove the whole thing, the bottom part here, to the left by one codon. And we end up over here, and then the EFG dissociates. That's a massive conformational change. We're moving the whole messenger RNA and the two transfer RNAs by one codon to the left. It's accompanied by a ratcheting action of the large and small subunits. They do this kind of action. And so it's really a massive type of conformational change that's occurring here, but very precise, so we don't lose that interaction between codon and anticodon. So that is a very amazing process, and it's accompanied by the hydrolysis of a molecule of GTP, as you can see here. Now, that means that the overall process now requires the hydrolysis of two GTP molecules. One to load the transfer RNA into the A site, the second to carry out this translocation step of the process. This then is just another picture of this whole thing that kind of incorporates all of this. We start up here with the P site filled, a transfer RNA in the exit site, the A site empty. We load the elongation factor TU with the transfer RNA into that A site. We hydrolyze the ATP or the GTP, and we align that new transfer RNA into the correct position. Carry out the peptide bond formation. The peptide moves from the P transfer RNA to the A transfer RNA. Then we carry out, we move the top in a little conformational change here. Then the EFG comes in, shoves the whole assembly to the left here by one codon, and hydrolyzes GTP in the process. So that's one complete cycle. For every amino acid that's attached, we burn two molecules of GTP. One there in the loading step, the second in that translocation step. So think now how many molecules of high energy are needed for each amino acid attached to a protein. Two equivalents are needed to load the amino acid onto the transfer RNA by the synthase. So that's two. Two more GTPs are needed here for the actual protein synthesis on the ribosome. A total of 4 high energy molecules or equivalents are needed per amino acid in the protein synthesis process, thinking of both the transfer RNA loading and the ribosome itself. So that's the overall type of process that's occurring there. Okay, now we're getting to the end. We get to these stop codons, and these stop codons, there's 3 of them that are on your genetic code table. They do not code for any transfer RNA molecule, but instead there's several release factors that are shaped a bit like a transfer RNA molecule. They can fit into this A site. They can recognize this codon here, the UAA stop codon, and when they recognize that, they cleave off the completed protein. It's released out there, as you can see, and that then causes the dissociation of the large and small subunits of the ribosome and the release of the messenger RNA. So these release factors recognize that stop codon and terminate the protein synthesis process. Okay, I think what I'll do here is show these last few slides on the antibiotics and go on and show another movie of the thing that I think shows the overall process. There's lots of antibiotics that affect protein synthesis. And it turns out there's enough specific differences between protein synthesis in bacteria and protein synthesis in our cells that people can take advantage of that to design specific antibiotics that will poison protein synthesis in the bacteria but not have any effect on mammalian protein synthesis. And this is an example of one that is not in that category, but here is a typical amino acyl transfer RNA molecule. And the transfer RNA goes that way. And the first base in the transfer RNA is an A, so that's the A base there. Here is the amino acid, the side chain, the amino group, the ester bond that we have here. And here is this antibiotic called pyrimycin. And this is a small molecule. It doesn't have the whole transfer RNA, but it has some similarity to the transfer RNA. It has the ribose ring. It has an adenine-like group, but it's got methyl groups on that nitrogen, whereas the normal A does not. It has what looks a little bit like a ester bond, but notice it's not an ester bond, it's an amide bond. It's got a nitrogen in there. It has an amino group. It has a side chain that looks a bit like a tyrosine, but is not quite. Okay? This is what pyrimycin does. It can get in, bind to the A site. The amino group can react with the transfer RNA at the P site and form a normal type of peptide bond. It can then move over to the P site, but it cannot be attacked by the next transfer RNA that comes in because this amide bond is less reactive than the ester bond that we have in the normal amino acid. So what the pyrimycin does is that it comes in, binds to the A site, reacts and forms an attachment to the peptide and moves over to the P site, then stops. It poisons further growth of the ribosome. So it stops the ribosome synthesis after one cycle. So that's what the pyrimycin does. And the pyrimycin has been used very extensively to understand various factors. Here then is a list of different types of antibiotics and what they do. Streptomycin is a very old antibiotic, and it's specific for bacteria, and it inhibits initiation and misreading of the messenger RNA, so that's one of the old classics of antibiotics. Tetracycline is, again, specific for bacteria. It binds to the 30S subunit, inhibits the binding of the amino acid transfer RNA. Chloramphenicol inhibits the peptidyl transferase activity of the 50S ribosomal subunit. So that's the peptide bond formation step, again in bacteria. Cyclohexamide, on the other hand, is specific for eukaryotes. It inhibits the translocation. Erythromycin binds to the 50S of bacteria. And pyrimycin that we just talked about is indicated here. It causes this premature chain termination by acting as an analog of that amino acyl transfer RNA. And it reacts with both the bacteria and the eukaryotes. So that's what that thing does. That's the end of that. So let me go back here, look at the other movie. Okay, so this is the overall process of protein synthesis now. You can see that the two subunits come together. Let me go so fast here. Let me start it over again. Okay. What do I do now? Okay, there we go. This is from Ramakrishnan's lab, by the way, this movie. So here are these initiation factors we talked about, binding the messenger RNA, the first formal methionine transfer RNA. We've loaded that on. Okay. Now both things are in there. Okay, that's one of the initiation factors that's leaving. Here comes in the transfer RNA into the A site with the TU protein. There it is, TU. We sample the codon, hydrolyze GTP, release that, it moves over. That's the whole recognition step. Then we bring in the EFG, and that comes in there, and it binds, pushes the whole thing over by one codon. See that? Bingo. Then the GTP is hydrolyzed, and that guy is going to leave. And now we're just going to repeat the process. So that's the next next transfer RNA that's coming in. Now it's really moving along. So one after another, the new transfer RNA comes in, peptide bond is formed, translocated, moves out. So that's the old. And then the amino acid comes out a tunnel in the backside of the large subunit of the ribosome. So there's the peptide coming out of that backside through that tunnel. So that's another very interesting phase in the process. And now there's the release factor. Comes in, binds to the A site, binds to the stop codon, and here's another release factor that comes in. We hydrolyze some GTP that I didn't mention, but then the whole thing falls apart. The protein is released, the two subunits fall apart, and we're done with the whole process. Okay, are there any questions on that one? Yeah. Yeah, let me just